Hi everyone, and welcome to this presentation on the biochemistry of amino acids. Today we are going to explore how these small molecules play massive roles in life, from metabolism and protein structure to even being found in space. Let's dive into what makes amino acids so important. Amino acids are often referred to as the building blocks of proteins, and for a good reason. Every protein in our body is made from chains of amino acids, and these proteins are responsible for almost every biological function. Amino acids also help with metabolism and cell signaling. They're classified based on their side chains, or R groups, which give each one unique properties. Here on the left, you can see all the essential amino acids. In nature, most of the amino acids found in proteins are what we call L-alpha amino acids. The L refers to the specific orientation of the molecule, and the alpha means the amino group is attached to the alpha carbon, which is right next to the carboxylic acid group, which you can see on the left side of the photo. There's another type, D amino acids, which are just like mirror images of the L-alpha amino acids. They're not used in making proteins in most organisms, but they do exist, especially in bacteria. For example, some bacterial cell walls include D amino acids, and interestingly enough, small amounts also appear in our nervous system and hormones. So while they're rare in proteins, D amino acids still have some important roles. Here in the photo, you can see how the ammonia is pretty much just flipped as if it was being seen through a mirror. Beyond building proteins, amino acids are essential in other vital processes. For instance, they help make urea, which the body uses to get rid of nitrogen. They also contribute to making heme, the oxygen-carrying part of hemoglobin in our blood. Amino acids are also the backbone for nucleic acids like DNA and RNA, and they're precursors for important hormones like dopamine and epinephrine, which help regulate mood and stress. These functions show how central amino acids are to life itself. Here we're going to talk about the classification of amino acids by R groups. So we classify amino acids based on the nature of their R groups, or the side chains that stick off the main backbone. There are 5 main categories. There's basic, like lysine and arginine, which have positive charges. Acidic, like aspartic acid, which have negative charges. Aromatic, like phenylalanine, with ring structures. Aliphatic, like alanine and valine, which are much more neutral and hydrophobic. And then there's sulfur-containing, like cysteine and methionine, which can form important bonds in protein folding. These classifications matter because they affect how proteins fold and function. And the folding and functioning of these proteins we'll get to a little bit later in this presentation. When amino acids link together, they form peptide bonds. These are covalent bonds between the amino group of one amino acid and the carboxyl group of another. What's interesting is that the peptide bond has a partial double bond character, which means it's not very flexible. However, this rigidity helps these proteins form stable shapes, which is critical for their function. If the protein can't fold properly, it usually can't do its job. Peptides are chains of amino acids, and we name them based on how many residues they have. For example, 2 amino acids form a dipeptide, 3 form a tripeptide, and so on. We always list the sequence starting from the amino terminal, that's the end with the free amine group, and go toward the carboxyl terminal. This order isn't random. It reflects the way proteins are built inside cells. This is a linear sequence, and it's called the primary structure of a protein. Amino acids have different functional groups, like the amino group amines, the carboxyl group containing a carboxylic acid, others like a hydroxide group, a thiol group, and even an imidazole. Each group can act like an acid or a base, depending on their pH. The pKa value tells us at what pH a group will be charged or neutral. This helps us predict the overall charge of an amino acid or protein at a given pH. And then there's the isoelectric point, or the pI, which is the pH where the amino acid has no net charge, and it is neutral. This concept is super useful in techniques like protein purification and even PCR experiments. In free amino acids, pKa values are pretty consistent, but inside a protein things can change. The local environment, like whether a residue is buried or exposed or surrounded by other charges, can shift those pKa values one way or another. This shift affects whether a group is protonated or not, which can influence things like enzyme activity, protein folding, or binding to other molecules. So the same amino acid can behave very differently inside a protein compared to its function on its own. So here we go, we're going to wrap it up. Amino acids are incredibly versatile. They're not just protein builders, they're also involved in metabolism, signaling, and even in origins of life studies. In fact, amino acids have been found on meteorites, which supports the idea that they may have played a role in the origin of life, which is very, very interesting. They also relate to health. Some plants produce toxic amino acid derivatives, which can affect humans and animals. So understanding the basic concepts of amino acids actually helps us in everything from medicine to space exploration. Here I'm going to test you guys on your concepts and understanding of the presentation given. So for the first multiple choice question, it's what type of amino acid is lysine? Is it acidic? Is it basic, aromatic, or sulfur containing? For the first question, it is B, basic. Number 2, which type of amino acid is primarily used in protein synthesis in living organisms? Is it A, D amino acids, B, L alpha amino acids, C, beta amino acids, or D, aromatic amino acids? In this case, it will be B, L alpha amino acids. And this is because D amino acids aren't typically found in most organisms, rather bacteria. And L alpha amino acids actually work with proteins and protein synthesis in living organisms. Number 3, what determines the isoelectric point, or pi, of an amino acid? Is it A, temperature, B, pKa values of its ionizable groups, C, protein structure, D, molecular weight? In this case, it is B, pKa values of its ionizable groups. For number 4, the question states, which metabolic compound is synthesized using amino acids? If we look back at the previous slide, the answer is B, urea. What characteristic of peptide bonds restricts protein chain flexibility? Is it A, hydrogen bonding, B, ionic attraction, C, partial double bond character, or D, disulfide bridges? If you guess C, you are correct. That's it for the presentation today. I just want to say thank you. I hope this presentation helped you understand why amino acids are so central to life and possibly beyond that. Thank you.